For over a century, the popular image of paleontology has been a process of cautious deduction, researchers extracting fossilized remains to infer the behavior of a living animal from a single static bone. That image is rooted in the work of 19th century pioneers like Richard Owen. While laying the groundwork, their methodologies locked our understanding of prehistory into strict, inflexible categories. This vintage illustration plate reflects an era where scientists split all dinosaurs strictly into bird-hipped or lizard-hipped groups. They defined them as cold-blooded swamp monsters and grouped entirely different lineages of saber-toothed predators together under the assumption that they shared the same heavy ambush tactics. Those 130-year-old assumptions are now being dismantled. Today, researchers deploy isotopic tracking to reconstruct prehistoric diets, process massive anatomical datasets through computational models, and sequence ancient genomes pulled directly from the ice. The textbook classifications many of us learned in school no longer hold up. Ancient ecosystems were filled with beasts far more biologically complex than early science ever pictured. Take the dinosaur family tree. For generations, the core tenet of the field was a strict division of all dinosaurs into two main branches, based entirely on the shape of their pelvis. In 2017, a study used computational power to analyze anatomical datasets across dozens of species, rather than relying on a single bone. The results challenged this foundation, suggesting that the most basic branches of the dinosaur family tree were incorrectly sorted. This visualization maps the results of that data model. When computers compared hundreds of skeletal data points, they found deep, unexpected links between supposedly distant groups, the theropods, like Tyrannosaurus, and the plant-eating ornithischians. The data highlighted highly specific shared traits. Both lineages independently developed an identical gap in their jaw structures for specialized teeth, and both showed physical evidence of early protofeathers. Based on this evidence, researchers proposed a new biological supergroup to house these linked animals, Ornithocylida. By prioritizing massive datasets over single anatomical features, scientists are finding that extreme traits, like hypercarnivory and feathers, evolved in ways that do not follow a simple, linear path. We see this exact kind of extreme biological adaptation in the late Cretaceous Prince Creek Formation. This region in ancient Alaska was an environment defined by freezing temperatures and months of total polar darkness. Under the old scientific dogma, cold-blooded reptiles were incapable of surviving these conditions, yet the fossil record reveals a thriving community of predators. This chart contrasts a standard 12-meter tyrannosaurid with a local predator, Nanuqsaurus. To survive the severe winter resource scarcity of the north, this animal shrank to a pygmy size of just 6 meters. The local Trelodon showed the opposite reaction. It grew significantly larger than its southern cousins to retain body heat and developed massive eyes for hunting in the polar night. Biologists call this Bergmann's rule, the principle that animals in colder climates grow larger to minimize heat loss. Finding active endothermic predators thriving in these polar forests suggests the biological limits of the dinosaur era were far wider than the traditional sluggish reptile model allowed. Moving to the Pleistocene epoch, we find another highly adopted predator, Homotherium. For decades, it was mislabeled as just another heavy, ambush-hunting saber-toothed cat similar to Smilodon. Looking at this skeleton, trace the distinct sloping spine from shoulder to hips. It's built like a dog, not a heavy cat. The forelegs are long and narrow, and the heel bone is extremely short. These are specific adaptations of an endurance runner. Chemical evidence confirms what the skeleton suggests. Isotopic analysis from fossil caves in Texas proves their diet consisted almost entirely of 2-year-old juvenile mammoths, which they selectively hunted across open steppes. The recovery of a mummified Homotherium cub from the Siberian permafrost provided a rare opportunity to analyze actual soft tissue and intact DNA. Genomic sequencing of that DNA revealed positive selection for genes related to respiration and daytime vision. It also showed cognitive markers strongly associated with complex social pack hunting behavior. The physical mummy also proved their massive teeth were completely hidden behind soft tissue lips, unlike the exposed fangs typically associated with Smilodon. Forensics transforms Homotherium from a generic monster into a highly specialized social endurance runner, perfectly adapted to its ecosystem. The rigid archetypes of the past, the sluggish reptiles, the solitary ambush predators, the neat evolutionary trees, have been challenged by a more nuanced reality. The ancient world was instead a highly dynamic, vibrant ecosystem, populated by feathered snow dwellers and marathon-running pack cats. By combining computational models, isotopes, and ancient genomes, researchers are no longer guessing at the shapes of bones, but documenting the biological mechanics of a lost world.